Welcome, Jason, to the Collaborative Research Center, Origin and Function of Meta-Organisms. You just gave a fantastic seminar to our community. You are a world-renowned expert in fungi genomics and genetics. You have a very strong background in computer sciences. Your papers are cited thousandfold. Some of the papers I just saw 1,800 times, an early Nature paper. And you develop tools to see the fungi, to see a world which so far, to us at least, is not yet so obvious. Thank you for the time that we are chatting a little bit about the background behind your science. And my first question usually goes to, you started your training with computer science at Duke University for your bachelor, but can you tell us, how did you get in that field? What did induce you to get there where you are right now? Was that whatever, your parents' garden, or how did you get where you are right now?
It's a great question, the origin question, for sure. I think when I started out, I was really interested in how systems work. And so when I was studying computer science, a lot of what I was focusing on how computer systems worked and how we build parts of it and put it together into kind of big networks or databases. And I got really interested in kind of information capture. So that was kind of like where the computational part of my brain fits in. I mean, I always, I grew up in the woods in North Carolina. And so I would go out in the backyard and play. I don't know that I had more of appreciation for trees than for the fungi. I think at the time, I don't think I really had as much of an understanding of all the organisms that were there. I just loved being outside. And so when I was a student, I worked actually in human genetics. I trying to use my computer work to help at the origins of some of the human genome work that was starting to become more available, and how do we integrate the different kinds of data, different kinds of questions. But then when I went to graduate school, I really wanted to kind of build on how to apply these tools to kind of data sets where we maybe didn't know as much about what's going on. And so to some extent, it was opportunistic. I had an advisor who worked in yeast genetics, and we talked about some of the data sets that were coming available. I worked in some C. elegans and Sina rabbititis lab as a rotation student at Cold Spring Harbor, actually. And so I learned a lot about just different tools. But then I came back to fungi because partly we didn't know as much about what they were doing. And I felt like yeast was this really interesting model system, but there were all these other types of fungi as I started to learn more. And, part of that was inspired by, at Duke, there's a really strong mycological community and training that there's just a lot of excitement. So I would say that part of my origin or my reason is that the community of people made me feel like I could contribute something and was welcomed. And not that other communities weren't welcoming, but this was just where I landed and felt such joy in kind of the discoveries that we could make and the people that were part of it. So for me, it was a lot about being able to be part of a group and a team and have a contribution. And so that was as strong as the organism for me. And then as you get into it, you realize there's so much we don't know, and there's so many different groups that there's new places to make discoveries. And so I felt like there's lots of room to maybe be a little bit naive to the beginning, because there's so much that was not known, and I could get in there and make a contribution. So that drove some of my choices at the early days. Now I'm in love with them, so it's not a problem. But
thank you so much. I think we come back to that at the very end when we consider maybe the junior generation right now, and this is certainly something important for them to learn, how to find your niche, how to find your place in a competitive world. So for our listeners here, could you summarize in 2-3 sentences your recent observations and discoveries? And you talked to us about the 100 Genome Projects, and could you summarize in a few sentences, if you think back in the last 5 years, the major contributions which came out from your lab?
Yeah, I think I guess that the driving force has been how genomic tools have led us to really understand the evolutionary relationships of fungi, of these organisms, and has revealed both sort of patterns in how they evolve that were surprising, and sort of how much the lens of genomics gives us as a way to kind of infer function. So we can start to connect, pathways and genes from model systems into non-model systems and see how the traits evolve with that. I mean, that's I think the driving hope with it. And so part of it is generating resources to make that possible, and then part of it is really digging into the data.
I think
at a high level.
Yeah, I think this is exactly this new world which allows now to take organisms into deep analysis, consideration, evolutionary context, which were out of the scope for a long time. I grew up in the developmental biology community in a world where we had maybe a handful of experimental organisms, the mouse, the fly, the worm maybe, and then there were a few others which struggled hard to get recognition. But now your new technologies make in essentially every organism a model organism. So how would you define a model organism? What is a model organism then? Isn't any organism a model organism?
It could be. I think you need those tools for being able to grow it and manipulate it. And so I think, yes, I think that distinction of what is a model probably should go away a little bit. It might just be, where do we have more resources, or more genetic lines, or a better understanding of the evolutionary history, diversity of the organisms. I think that it is breaking down that, especially if technologies like CRISPR allow us a lot more kind of finesse and being able to create genetic backgrounds that we can test hypotheses in. So I think, depending on your question, right, any system can be a model as long as you can make it reproducible, and that you have the ability to really control the growth or control the experiment. And so, there are fungi like arbuscular mycorrhizal fungi, which we can't grow for the most part as pure cultures, right? They're part of a community system. So it's hard to absolutely have reproducible research on every round, because there's some variation in how we get the strains. But that's okay. I think that's what the tools allowed us to measure what those differences are, and then we can maybe use that in our understandings, interpretations of it.
Yeah. People may wonder now what the tools really are, what he's talking about. Is that just sequencing? I mean, thinking back on your early work, you were deep in computer languages, etc. Can you summarize a little bit? So what are the new tools which you have developed, which you're sharing now with the worldwide community, which is so fantastic, and which then also brought us here to northern parts of Germany. So that's what is the technology really which you're using now?
Sure. So I think there's a couple of dimensions. So one is the inexpensive sequencing has driven so much of the field now. I mean, you have to have good questions to do the research, but the technology has let us ask those questions in so many more labs, right? The cost of doing the work is now accessible to more, but not everybody yet. I do want to acknowledge it is still expensive, but it is much more approachable. And so the ability to generate high quality deep sequencing, as well as now long read sequencing that allows us to really put together genomes or transcriptomes accurately. That's a fundamental kind of useful resource that we can build off of. Now, the other component is, how do we interpret that data, right? Just having it doesn't get you anywhere. And so I have benefited from these, incredible resources and research that have generated, really fast algorithms to align sequences or annotate sequences. And so we've put together packages that kind of build on these individual tools to, for example, make it easy to run a genome assembly kind of in at least an automated fashion for what we need to do, or run a genome annotation, so predict where the genes are. It's with fungi and eukaryotes, you have some more complications with introns and whatnot. So we have a lot of effort to try to make that as straightforward as possible, but using best practices. So those are kind of like components to that. And then I guess the phylogenetic analyses that allow us to infer the relationships between organisms or between genes based on sequences. Again, the developers of those tools have made them faster and taken advantage of the faster computer processors. And so now we can really process large amounts of data in a reasonable time frame. So we've caught up on both sides, right? We've been able to produce more data, but we also have the technology, the computational technology, and the algorithms that can kind of properly process all of that so that it is feasible. I think that the other component, of course, is just the biological resources, having cultures, having herbaria that have collected these organisms and know where to find them if we need to go get a fresh sample. There's been natural history that's developed that helps us understand the context of these organisms and where to look for them if we need to get new ones, or maybe go look at a specimen. And so both the living culture collections, which have amassed over more than 100 years of research in some cases, are also really critical, because we want to compare organisms. We need to have someone have defined what those different organisms are, and that those are readily available. So I think we're really lucky. I feel very lucky to be in a field and in a time where these different resources are all available, and then it's just about trying to put them together and to think logically about how we can connect those results. So that would be that. And then I guess just one other component would be like the sequencing technology and the PCR-based technologies to go out and sort of look at an environment. So we can also look at things that aren't cultured and sort of sampled in the environment. And again, having this really deep sequencing approaches allows us to go in and do molecular ecology, where we can actually look at the microbes from a sequence-based approach and not even have cultured them. So yeah, I see a lot of what I do is putting pieces together, but having an idea of how this technology plus this algorithm or this computational approach can help us solve or answer a question.
Yeah, isn't another component still is the visualization of the data? I mean, we have to, you collect enormous amounts of data. Then you have now programs to interpret them, but you also have to make the data kind of interpretable by visualization. And I saw in your slides, I mean, there are wonderful ways now to make it kind of comprehensive to a person who really doesn't understand the details of where the data come from. But if you show these complex trees, oh yeah, sure, this is related to that, and they are different from them. Yeah, great. Extending on what you said just a minute ago, I think if you think back in the old days of mycology and the fungi experts, who were these scholars? These were ladies and gentlemen, and they had deep insight in the biology, also probably the physiology, the histology of certain fungi. Now, today, you are mathematician, computer scientist, whatever you're called, and are geneticists, then there are the biologists, then there are the modelers. It's multidisciplinary. We live in a very different world. Isn't that a challenge? Do the mycologists understand what you say? And do you understand what they say when they talk about a very specific physiological feature of some fungi living in the desert or living somewhere? How do you handle that multi-language community, which is the only way to move forward, of course? Because the old-fashioned mycology that didn't tell us how important fungi really are. Your technology, together with them, tells us, oh my God, fungi are so important, we have to look carefully. So how to tackle the multidisciplinarity challenge?
That's a great question, and I think a great thought about how to prepare yourself for this is, so So it is hard to speak multiple languages, as you say, and I think how we work collaboratively is really important in making that effective. So I think that I won't say that I'm an expert in all of those things, right? I feel comfortable in much of mycology, but there are certainly very specific terms and very specific processes that I don't find myself familiar with or able to speak authoritatively on. However, I feel like I have been equipped with the ability to learn and to speak with those. And so basically being able to rely on in an interdisciplinary way by trusting and relying on other experts to synthesize that. Now, it does take some practice to be able to talk about the kind of nuance of how we do high performance computing and set up our analyses and think about proper ecological design in our ecological experiment and maybe statistical proper statistical interpretation of the results. It's hard to do all of those at once, and so I think that having a team and being able to rely on each other for that is really important. So that's why these kind of collaborative either research centers or research teams, it's just really important to see the value in what each member brings to that. But it does mean getting outside your comfort zone. So I think for a lot of my career, I've always felt like, there's a bunch of this that I don't know, and I'm going to try my best to understand it so I can speak with those that are experts and learn from them and try to build. And so it's something that you maybe can't do all at once. You have to kind of bite off a chunk of that and try to get better at it. So part of our work is I didn't speak about, but is medical mycology too. So understanding how fungi interact in typically in a human host. And so I don't understand necessarily all of the human immunology components to some of these problems, but I can rely on the team members to help focus on that question, and I can bring my component there. So I guess, in terms of the future of science or the future of these kinds of research, it's really being able to trust and develop a team approach to the science and informing yourself enough that you know what the other members of the team are doing, even if you can't really do all of their work. So I think that's, there's trust and because you want to write, you want to produce results that are truly reproducible. And so you really need to trust your team about when something is put together. But I think it's also about kind of being willing to be humble and know that you don't know enough about something and to try to really dig in and learn from somebody about that. So I think that's how I try to approach my collaborations and how I've been able to learn, I guess, over the years. But
I think this is a great statement that it needs a great deal of trust and confidence. You're having the right team players, and then you go on with your expertise. That is, I fully agree, and probably many people, including us, we try to see this challenge in the same way. This is the communication within the scientific community. But you just said, and this is of course true, that fungi are of tremendous medical importance. And when it comes to health and disease, there's another part, and that is the general public, which needs to know what's going on. And I know I saw that you are very active and also quite successful in what we call public outreach and talking in simple words to the public. Isn't that also becoming more and more important, that we find not only a language to talk to our team player from the different discipline, but also to the general public, because not only that they have the right to know what we are doing, they trust what we are doing, but also they need to know what you just observed, because it may be important for them. I bring that up because, again, when I grew up, talking to the public and to the media, not to speak about all these social media activities privately, was of course a no-go. And it was a no-go for a hardcore scientist to do too many interviews and media interactions. I think this the time has changed, and it is important to do that because of the reasons I just mentioned. What do you think, Jason?
Yeah, I mean, I think it's really critical to spread the knowledge that we're building. And I think, I mean, I take pride in being a professor and being able to teach and to be able to see part of my job as educating. And so I feel that doesn't need to stop at the classroom. If I have a chance to provide knowledge, broader, I think I need to do that. I think in particular with something like emerging infectious diseases, where we want to communicate facts, right? What do people need to worry about or not worry about when it comes to a particular fungal disease, let's say? We want people to hear facts and know how to react to that and know how to use that. So I think it's really important that information is coming from those that understand the facts. And so I think if scientists aren't willing to speak about it, then who is, right? And they might take a different perspective on it. And I mean, obviously, it's hard to make some of what we do exciting and interesting to those that aren't specialists. So there's a challenge in how to communicate and keep the interest in what we're doing if you're getting very detailed. So there is some practice, I think, in how to distill the important parts out, and that's part of being an educator, is making sure I can hopefully keep the interest in what we're doing to our audience. And so I do feel like we have a responsibility to communicate what we're doing. Even if it's not public health related, how do these research studies inform the climate crisis? How do these research studies inform, different ways that we might develop renewable technologies? I think there are all kinds of ways that fungi can play a role in that. And so if we can get some other members of the public or other students excited about working in this, that it's almost like it's also important to recruit, right? It's important to get more possible people excited about it, because they might come up with an idea and a contribution. So I'm a big believer that of a kind of the big tent approach to some of the science. I would like more people there to help us think about the problems, and that there's a diversity of ideas in there, because without that, we may not think of or come up with the right way to solve some of these problems. So yeah. I think there's both the kind of mycology is a small field, and I would like to see it stay strong and get bigger. And so the only way I can see that happening is if we convince more people to keep studying it. And so I hope that we can inspire excitement around that, in addition to how this might help obviously have practical implications on health or medicine or agriculture. So
very, very true. Let's stay a moment in your classroom, and you are entering it, and then the young students sitting there, they wonder, will I ever make it in a scientific career? And they may ask you, you are very well established, successful, visible, international visible, you made it. What would you tell, what would you want to tell them, what's important at the very early days, and then maybe during their undergraduate training, but what's important for pursuing a scientific career when you come from school, so in the early days? I mean, you mentioned already in the beginning, you shared with us your personal motivations and so on. So what would you tell, or what do you tell your students in your classroom? What does it take to become a successful scientist?
Yeah. I mean, first, there's not one prescription, thankfully, but I think. And again, I know that luck plays a role in everybody's success, and so I think that picking topics that are that there's an opportunity to make a contribution to was important to me. So I kind of mentioned I guess I felt like the field wasn't competitive in a way that I could come in and be somebody new to the scene and provide something to it. So I think I looked at where there were opportunities to some extent. I mean, it wasn't so planned out. It was more, this is really interesting to me, and it looks like I could make a contribution. So I want to dig in and learn more about this and then get excited about it. So I mean, I think for a student, there's obviously other things they can't control, right? It's hard to control, whether or not there's lots of funding in your field, or whether or not there's lots of competitiveness, or openness to new ideas. That you can't always control that. But so I don't want to be too rosy about that. If you do these things, it'll all work out. But I do think that interest and excitement in your questions are really important, because you need to have that intrinsic motivation to keep going on a project, or because there are going to be really dark times, or at least hard times, when it's really frustrating, or you're not sure why you're doing what you're doing, and you need to have something that's going to keep you going through those periods. And hopefully that's because you like what you're doing, or you see an end product that you like, that you'd like to know the answer to. And I think that's probably for me, that's been helpful, and I think I've seen it in others that have been successful, is that there's an intrinsic, excitement or desire to understand it. Now, that doesn't necessarily have to be organismal. It could be that you're really interested in the problem space of, how to solve these computational problems, and maybe different types of data are really cool, really interesting ways to kind of bring that together. I think it's important to find things that you really get, interested in. I think the other part is a willingness to, for me anyways, was a willingness to be flexible, in that, it wasn't all planned out that I had to work on this particular one organism. I really wanted to look at a couple of different ideas. And I mean, you mentioned, I have a lot of collaborations and a chance to work with different people. It's really exciting. It's also it's also hard, because you're maybe not an expert in all of those systems always, and so you have to kind of feel comfortable being, having to do a lot of extra learning, maybe to get competent at some of those parts. So just in terms of some of the sub parts of the field. So I think that for me, being versatile was really important. I brought computational skills, and so and that allowed me to work in different places. And those computational questions and approaches were really could be applied in multiple ways. So for me, that was helpful. And so I try to, at least students that come out of my group, I really encourage them to build mastery in computational, understanding about data processing, about how to automate things, and how to really, build these kind of fundamental skills, because they can be applied to lots of different, even non-biological questions. And so there's a career for them, even if they decide that that's not, they don't want to do a scientific or a biological sciences. There's lots of opportunities with those skills. So that's what I think there's a benefit there. But at the same time, there's a real benefit in narrowing down and focusing on a particular organism or a particular set of questions, so that you have that expertise. I mean, I think you have to be an expert in something in order to be successful too. So I think there's some trade-offs in terms of being too broad and too shallow if you don't spend enough time on a thing. And so I guess that's, how to find that balance really depends on the individual. But I think those are some of the, I don't know, in a nutshell, those are some of the ways I've kind of approached it.
Great answer, very balanced answer. I think, I guess we all agree with all the points you made. We are unfortunately coming to an end of this interview, but before that, two last questions, and we return back to the field. And the one question I want to get an answer from you is, what is the major open question in your field right now? And the last question then is, and where will the field be in 5 years? So what's the major question? What is the open question? The major open question in the fungi kingdom field?
Oh my gosh, there's so many great questions to be asked there. It's hard to pick the one. I think for me, how fungi interact with other microbes and with hosts is really, I think it's driving a lot of the questions that we're trying to answer, and the field is trying to answer. How do they manipulate their environment and their interactions with those hosts to succeed? And that's there's so many different facets to that. How do they detect and know that they're interacting with a host that they want to make a relationship with, if it's a plant system, or how are they detecting the signals to colonize if it's an animal host? And so I think all of that is just really dissecting that from the molecules that are the fundamental communication between the microbe and another organism, another microbe. And so I think that the technologies of metabolomics and sort of more high density kind of sampling of these chemicals and these communications is going to be part of that too. So how we kind of integrate that up through the genetic underpinnings of those networks and the organisms themselves, and then how that really interacts with how that modulates those interactions between the organisms. So I think for me, that's where I think that the fields are going to be kind of converging on really dissecting those different parts. We've done a lot of the work has been genetic, and I think now we're trying to understand even further those products that mediate those interactions. And then, because I'm an evolutionary biologist too, I think all of that's in the lens of how has this occurred? How have these kind of adaptations and these interactions that have kind of maybe have their optimizations, I guess, right? Their preferences for who they like to interact with. How is that pattern repeated in different parts of the evolutionary history of the organisms? And does that tell us something about the kind of fundamental processes and the fundamentals of that? So I think that's, it's very integrative, but in terms of, but I think those are the big questions of how we kind of integrate from these small interactions and small components up to whole organisms, and then are those organisms sort of adapting evolutionarily in the same way? Are there different things we can learn about that? Because that's going to inform, practically how we develop, strategies for fungal applications, and how we use those in an agricultural field versus the concerns that we have spillover into problems that cause disease, have organisms that are hard to treat in the clinic. And so I think that those are what we might use that understanding for as well.
Last question: will there be intelligent translational products in 5 years?
From basically, like, I can predict something from a genome and use that in the clinic, or use that in the field,
or your volatile compounds. I mean,
Yeah, I'd like to think that we'll have more understanding of how to sort of develop, if it's like a blend of different compounds that really induce and infect in a fungus. I think that in 5 years, I think there'll be parts of that we will understand better. Whether or not we can sort of screen a metagenome and pick out the components from bioinformatically, and then turn that into something that we synthesize and it's going to work. I think we're a bit further from the predictive, being able to make predictions, but I think we can narrow candidates down with a lot of the technologies and then use that to develop testable systems. So I hope so. I hope there'll be some more predictive.
That's a wonderful perspective, of course, and telling us that we are really watching now with amazement just an emerging field which is going very quickly along the path. I would like to thank you, Jason, for sharing with us your really personal views. I really appreciate, and all the Collaborative Research Center and the listeners of this program appreciate when persons share really views which are not in a high glossy scientific article, but this is essential to understand how did the glossy article then become reality. Thank you so much for being here, for spending some time with us, for sharing your views, and very much looking forward to see you not in the online format, but in reality here in North Germany, in the Collaborative Research Center, or somewhere else in the world. Thank you so much for coming, Jason.
Yes, thank you very much for the invitation. Thank you.